Okay, thank you. Hello, my name is Marina Luz Ingravidi. I am a PhD student at the University of Buenos Aires in Argentina, and I will be presenting the poster titled Genomic Analysis of Fmr1 Cluster, Possible Implications in Fragile X Associated Diseases. The Fragile X messenger ribonucleoprotein 1, or Fmr1, is a 38 kilobases gene located in the long arm of the X chromosome. On its 5' untranslated region, it presents CGG repeats of variable length. The length of these repeats is associated with various diseases. From 10 to 54 repeats, the alleles are considered normal. Between 55 and 200 repeats, the alleles are premutated, and this premutation is associated with 3 diseases: FAXTAS, FXPOI, and FXAND. And when the allele has more than 200 repeats, it is considered full mutation, causing Fragile X syndrome. Fmr1 encodes for the synaptic functional regulator Fmr1 protein, or FMRP. This protein is an RNA binding protein mostly expressed in the brain and the testes. The microRNAs are a small, mostly post-transcriptional genetic regulators that bind to mRNA by a region called seed that includes the nucleotides 2 to 7. And the dysregulation of these microRNAs is implicated in many diseases. Recently, a microRNA cluster was described in human and mouse adjacent to the Fmr1 gene and was named Fmr. In human, the cluster contains 22 microRNAs, and it was found that some of them regulate Fmr1 expression. In our lab, we have been using the rat as a model, and thus we were interested in describing the cluster in this species. We established 2 main objectives. First, to perform a comparative genomics analysis of the Fmr region in Homo sapiens and Ratus norvegicus. In particular, to identify the orthologous region of the Homo sapiens cluster in the genome of Ratus norvegicus, using the NCBI genome browser to determine if there is structural conservation between human and rat. Secondly, to generate a genomic map of both Fmr regions, obtaining the sequence, the ID, and the position for each microRNA in the cluster from 2 different databases, MIRBase and MIRGeneDB. And finally, to study the sequence identity between the Fmr region of Homo sapiens and Ratus norvegicus, identifying orthologous microRNAs using sSearch, an in-house pipeline, and the software Mauber. And our second objective was to identify the microRNAs present in chromosome X that potentially target Fmr1. In particular, to identify all the microRNAs in chromosome X that potentially target Fmr1, and to search for all of the targets of each microRNA found in the previous step. All the target predictions were made with MIRDB. As a first result, we found that the rat's Fmr cluster contains 18 microRNAs flanked by the genes SLIT-RK1 and Fmr1, and are located in the same transcription sense, as we can see in figure 2. We also observed that many microRNAs from both species belong to the MIR506 family, suggesting a common origin. We then performed the sequence identity analysis, and we found 6 conserved microRNAs between human and rat Fmr, as you can see in table 1. Then, with the software Mauber, we generated similarity profiles and observed that only 4 of the 18 microRNAs are located in conserved regions. And finally, we found 9 microRNAs predicted to target Fmr1 in rat. One of them, MIR883P, is located in the cluster, and it is known to regulate Fmr1 in the mouse. In table 3, we can see that the position of the microRNAs that may target Fmr1 relative to that gene is variable. In brief, we found that the cluster Fmr is evolutionarily and structurally conserved between Homo sapiens and Ratus norvegicus, most of the microRNAs belonging to the MIR506 family. Surprisingly, we also found a big divergence when it comes to nucleotide sequences. And regarding the target search, we can conclude that there is 1 microRNA in the rats cluster, MIR883B, that can potentially regulate Fmr1, although it has no orthologous microRNA in human. Thank you very much.
Thank you, Marina Luz. If we have questions, please, you have Q&A to make them. I have a question. Have you made gene ontology, for example? Did you study which processes, biological processes, are regulated by these mRNAs?
You mean the cluster? The
Yes.
MRS? No, it's something that we will be performing during my PhD. This was part of my another work that we did while I was an undergraduate student. So for my PhD, we intend to see the MIR506 family, in which processes they are involved, these microRNAs.
All right. And which other experiments are you planning so far to study this cluster?
We intend to, on one hand, to add more target prediction software to like have more robust information, and also with the targets that we found, the microRNAs that may potentially target Fmr1, we intend to evaluate them, analyze their expression in the tissues that we the tissues of interest, that is the ovary, the rat ovary. We intend to evaluate them, the expression and their location.
All right.
That's
I think we have a perfect.